Okay, welcome to a symposium session on HIV drug resistance and antimicrobial resistance. This is a very big auditorium, so we're going to ask, so we look more like a mass of people. If you want to fill in down front, that would be very helpful. My name is Meg Doherty, I'm from WHO, and I'm happy to be co-chairing with.
Hello, everyone. I'm Avlina Gokeng from Cameroon. I'm virologist and I work for the Institute for Research and Development and the NRS, which is also part of this conference.
So to start out, we thought because many of you may or may not have been part of the HIV drug resistance satellite that happened yesterday, that we could begin the whole session where we're trying to examine how HIV drug resistance links into the broader drug resistance environment, AMR, tuberculosis, as well as malaria resistance. That's the theme of this afternoon's speeches and discussions.
It's my pleasure to introduce our first speaker this afternoon, which is Dr. Santiago Avila from Mexico. Dr. Santiago Avila is a researcher at the Center for Research in Infectious Disease of the National Institute of Respiratory Diseases in Mexico City. He received his MRC degree from the University of Nottingham and his PhD from the National Autonomous University of Mexico. He has conducted numerous researches on molecular epidemiology, transmitted drug resistance surveillance, HLA-associated evolution, and in different regions. He coordinates the surveillance and participation of the institution as a WHO reference national laboratory in the region. He coordinates the molecular biology laboratory, and he is co-coordinator of the Mesoamerica project, a collaborative study involving investigators from Mexico, Guatemala, Honduras, El Salvador, Nicaragua, and many other regions. Then please welcome Dr. Santiago.
Thank you very much, and thank you for the introduction and for allowing me to speak here and show you some of the results that we've had surveilling transmitted drug resistance in our region. And it's my role here to introduce you to some of the new documents that were just released by the WHO. And so to start my talk, I'm going to tell you a little bit about these documents and tell you some of the results that they got from them. And also, I'm going to then focus to Mexico and tell you a little bit about what the national program is doing regarding the results of these documents that we found. So the WHO did this meta-analysis involving several studies outside the WHO guideline surveillance, and they found that transmitted drug resistance is a global problem. As you can see here, the prevalence of transmitted drug resistance to NNRTIs, in particular to efavirenz and nevirapine, is growing up in all parts of the world. And this is especially marked in Eastern Africa and in Southern Africa, as you can see here. And in response to this, the WHO has launched a global action plan on HIV drug resistance. And this provides a 5-year framework for a global response to HIV drug resistance. And it's based on surveillance of HIV drug resistance in several countries. In this case, I'm showing you the plan for responding to pre-treatment drug resistance. That means resistance before people start antiretroviral therapy. When there are no data available, the WHO strongly recommends to implement these nationally representative surveys to gather information on drug resistance in each of the countries. And when there is information on transmitted drug resistance at the national level, you can see that the WHO established a threshold for action. So if resistance to efavirenz and nevirapine is over 10%, then action is strongly recommended. And so the main thing that countries can do is change to non-NNRTI-based first-line regimens, for example, those including dolutegravir. For places where this is not feasible, maybe implementing drug resistance testing before starting ART. WHO pre-treatment drug resistance surveys give national estimates of transmitted drug resistance, well, pre-treatment drug resistance in this case, for 3 groups. First, for all antiretroviral treatment initiators. That includes people who are naive, who have never been exposed to antiretroviral treatment, and also for people who have had this previous exposure to antiretroviral treatment and for some reason have stopped and then restart antiretroviral treatment with first-line regimens. And several countries have already started implementing these surveys, these pre-treatment drug resistance surveys, and 11 of them have already results available, and I'm going to show you these results. 4 countries in Africa, 6 countries in the Latin American region, and 1 in Asia have already implemented pre-treatment drug resistance surveys with national representativity. And what you can see here is that the level of NNRTI resistance, that is resistance to efavirenz and nevirapine, are over the 10% threshold in several of the countries that have implemented these surveys. And so this is a very important thing to note. This is something that can threat the achievement of the WHO goals to arrive to the 1990 goals. Also, people who have been pre-exposed to antiretroviral treatment have shown to have much higher levels of NNRTI resistance than naive people. And as you can see here, that has been seen in several countries, more or less 2-fold higher drug resistance for this group of people, and even much higher for some countries like Nicaragua, for example. Also, although there are no information specific on key population or special populations, what we can see is that there are some key populations that can be more affected by pretreatment drug resistance. And here you see the example of women versus men. And for example, in countries in Latin America, like Nicaragua, Guatemala, and Mexico, you can see a higher affectation in women compared to men, and that has to be taken into account for global response. And so where do we go from here with these results? So the WHO has implemented 5 points in the global action plan that you can see here in purple. And so this is what has been discussed at the regional level regarding prevention and response. We need to strengthen health systems, especially with viral load monitoring to identify and promptly manage viral failure. Also, prevention programs are needed, including some more affected populations like people with prior exposure to antiretroviral therapy, or also women in some settings. And of course, education on antiretroviral prescription by physicians. And regarding monitoring and surveillance, and also research and innovation, there's a need to assess pretreatment drug resistance levels in countries that lack this information. Although you saw results for some countries, there are several countries still lacking this information, and it's very important that each country generates their own data. And the data currently being generated can be used to convince national programs from other countries that this is indeed an important thing. And also successful surveys can guide the implementation of new surveys. Also, international collaborations can provide support in survey design, logistics, HIV genotyping, sample transportation. In our experience in Central America, it has been very important to involve academic institutions as well that can coordinate efforts to train people, to inform clinicians, and to design the logistics and sample transportation methods that are going to be implemented in the surveys. Also, regarding laboratory capacity and governance, when the levels of NNRTI drug resistance are high, we need to identify what response or action is needed or would be more suitable for each setting in particular. The change to dolutegravir-based regimens in all ART initiators is the preferred option, but we need to see if it's suitable to implement this action in all the countries. Also, we need to prioritize dolutegravir-based regimens in individuals at high risk of PDR, like people with prior exposure, or perform baseline HIV drug resistance tests in the case where a switch to dolutegravir is not feasible. These are the results of the PDR survey in Mexico. As you saw, we have still not crossed the 10% threshold for PDR, for NNRTI PDR, but we already have levels over 10% for any antiretroviral treatment. And this is mostly associated with efavirenz. Mexico uses first-line regimens that are mainly based on efavirenz. And this, as you can see, K103N is the mutation with the highest frequency, as we have seen in many other countries. Women are more affected than men. And so looking at these results, even though when we have not still passed the 10% threshold, we really need to do something to stop resistance from becoming a real problem in the country. So what has the Ministry of Health said about these results and what's being implemented in Mexico? So we have a very big problem that the prices of dolutegravir are extremely high in Mexico. So implementing first-line regimens using dolutegravir in Mexico is not feasible right now. So our preferred option is to implement drug resistance testing for everyone starting antiretroviral treatment. In our case specifically, lab capacity exists, and doctors are usually trained on drug resistance interpretation because drug resistance testing is a standard of care in patients failing antiretroviral treatment. And an alternative option for us would be to switch to dolutegravir-based regimens. But the prices right now don't allow it. Price negotiations and licensing discussions are urgent and are taking place right now. Hopefully we'll get to a point where dolutegravir can be included in the first-line regimens. And also there's the option of considering other integrase inhibitors that can have prices similar to a tripla and other first-line regimens. Also, in a large and complex country such as Mexico, there may be other options like diversifying the response based on regional drug resistance prevalence. And so we have already planned our subnational PDR survey that will give us drug resistance estimates in 8 regions of Mexico with regional representativity. And this would allow us to identify places within the country that are more in need of an immediate action, such as implementing baseline drug resistance testing, or prioritize the use of dolutegravir-based first-line regimens, or reinforce viral load monitoring. In Mexico, usually people who are restarting antiretroviral treatment can be started on PI-containing regimens. And these are some data that follow up on the national PDR study in Mexico. We have already assessed drug resistance in 3 different places in Mexico. The northern part is the city of Tijuana. It's a border city that has a very complicated epidemic because it includes many risk groups, including MSM, people who are sex workers, people who inject drugs, migrants, etc. The central part where Mexico City is, and the one in the Yucatan Peninsula, that is Cancun, is a touristic center. And we can see that there are differences in prevalence of resistance in these three areas. And as you can see, resistance to NNRTI is increasing in all the regions, and this is because of the Faberens resistance and the K103 mutation, as you can see in the graphs on the left. What are the potential actions for this? Is that the change to dolutegravir-based regimens are costly. It's about 100 times more than what dolutegravir costs in Africa or in Brazil. And there's also an increasing number of persons in need of ART and a limited growth of available resources, and that limits the use of dolutegravir in our setting. The bureaucracy and administrative barriers that restrict drug availability in our countries. It takes a long time for a new drug to be approved to be used at the government. Also, price negotiations can be hindered by national laws and international treaties, or at least it can take a lot of time for drugs to be approved. And of course, the interests of pharmaceutical companies. For baseline HIV genotyping, laboratory capacity and human resources are a main issue. We need to increase laboratory capacity and training people for doing drug resistance testing. Also training doctors in the interpretation of drug resistance testing and acting upon them. Shortening the time to deliver results, that's a very big issue regarding baseline HIV genotyping, and also reducing the costs of the test itself, maybe using next generation sequencing. And finally, using modeling for decision making would be preferable, would be the best scenario. However, it's a very specialized area, and there are few people able or available to do this, and sometimes it takes a long time to develop models. So in conclusion, NNRTI PDR is around 10% in many of the low and medium income countries that have implemented surveys. Countries need to act upon high levels of HIV drug resistance in order to achieve national and global goals to control the HIV epidemic. There's a need to implement HIV drug resistance surveillance in countries where information is still lacking, and also here it's very important to tell the success stories of places where these surveys have already been implemented. And responding to HIV might be highly dependent on national resources and scenarios. I told you about Mexico and how different this scenario can be compared to some African countries, for example, or even some other Latin American countries like Brazil. The use of dolutegravir in all ART starters is dependent on drug costs, negotiations, and licensing. Also, baseline genotyping in all starters might be an alternative for some countries in our region, but we need to increase laboratory capacity. And alternative solutions, such as prioritizing responses based on subnational surveys, can also be considered. And with that, I finish, and I just want to acknowledge the people from our institution, the INER, people from the national programs, CENSIDA, the CDC, the Universidad del Valle de Guatemala, that's guiding the implementation of surveys in Central America, the PAHO, and of course the people from WHO who have done an incredible work putting together all this data and releasing these materials to guide the fight against HIV drug resistance. Thank you very much.
Thank you, Dr. Santiago. Then, as we will having a roundtable at the end of the different presentations, then we have the opportunity to take few questions before the roundtable. Then, to respond to Dr. Santiago's needs of modeling work, then it's my pleasure to introduce an expert in modeling. Then, Professor Andrew Phillips is epidemiologist with a high experience in HIV cohort studies and clinical trial, as well as cost-effective modeling. And it's a pleasure to introduce him for the next presentation. Welcome, Professor Andrew.
Thank you. Good afternoon. So I'm giving this presentation on behalf of this working group, and I'll give you the names of the working group at the end. So the background to this is that, as Santiago has just mentioned, the WHO recommends drug resistance surveillance in people initiating antiretroviral treatment. If high levels of pre-ART, NNRTI resistance are identified, options for ART program response have to be considered. And again, as just mentioned, potential policy options include transition of the first-line regimen in new initiators from efavirenz to dolutegravir, or introduction of individual-level drug resistance testing in people starting ART. Oh, okay. I've only just realised that I'm the only one who can see my slides. But I'm reading them, so just listen and you're fine. There's no pictures. Modelling can inform the most cost-effective choice. Oh, there we go. So the objective of this presentation is to evaluate the effectiveness and cost-effectiveness of policy options in the presence of pre-ART NNRTI resistance, and this is in the context of sub-Saharan Africa. So the policy options that are being considered in this modelling analysis are to have no change in policy, so continuing with a efavirenz-based first-line regimen in new initiators. Secondly, for all ART initiators, and this includes people coming back to care, reinitiators, to make that clear, having the resistance test at treatment initiation, and then use dolutegravir if NNRTI resistance is detected. And thirdly, this transition of everyone who's initiating treatment or reinitiating to start with dolutegravir. So those are the three main options considered. And the modelling approach is to use a model that's been used for has been used previously by our group. It's an individual-based simulation model of HIV transmission, the effect of antiretroviral treatment, and it considers the effects of specific drugs and specific resistance mutations. And the model's based around data so the model's informed by data from Europe and from Africa. But the actual settings that are recreated with the model are based around Southern Africa. And we generate what we call multiple setting scenarios through simulation, computer simulation. So we vary parameters such as the ART adherence profile in a population, the ART interruption rate, the switch rate after first-line failure, and several other parameters as well, independently at the beginning of each simulation run. So they're varied randomly within plausible bounds for settings in the region. And that allows us to generate different setting scenarios. And this gives you the characteristics of the 5,000 setting scenarios that we generated in this way. And I'm just giving you the characteristics in terms of some of the variables, and I'm giving you the median in the 90% range. And so we're looking at, I'm not going to read it all out, but you can see that we have a range of HIV prevalence, of HIV incidence, the proportion of HIV positive people diagnosed, the proportion of all HIV positive people on ART, the proportion of ART experienced people who have started second line, of people on ART, the proportion with viral load less than 1,000, and then of ART initiators, and this is ART naive initiators now, the percent resistance is a median of 10% in majority virus. And of the ART initiators as a whole, 18% on average had prior ARV exposure. So those are the sort of model data that arise as a result of that simulation procedure that I explained. So then to continue then with the modelling approach, for each one of those 5,000 setting scenarios, we consider the situation with the level of pre-ART N and RTI drug resistance that arose in that simulation. And we compare the outcomes of those 3 different potential policy options, projecting forward over the next 20 years from 2018 to 2038. And the specific assumptions on the effectiveness of dolutegravir versus efavirenz are informed by some of the studies below and others. As we know, there's a much lower rate of resistance acquisition with dolutegravir than efavirenz. There's also almost certainly a higher potency and greater tolerability. For the cost-effectiveness analysis, we take a health systems perspective. We discount the costs and the DALYs, our measure of outcomes is DALYs, disability-adjusted life years. They're discounted at 3% per year, and we use a cost-effectiveness threshold thought to be relevant for low-income settings in this region, certainly for HIV interventions, $500 per DALY averted. If you're talking more generally outside HIV, that's probably quite high. Absolute costs and DALYs are relevant for a country of the population of the size of about 10 million adults in 2017, such as, for example, Malawi, similar to that population size. So these are the simulated outcomes then. So this, and I've only got time to show summarize the results really. So this shows the comparison. I'm now restricting to setting scenarios where 10% of ART initiators in 2017 had NNRTI resistance, more than 10%. And this is showing the mean percent with viral load suppression on average over the next 20 years. And so this is averaged over those scenarios with more than 10% resistance. And you can see the predicted benefit of the pre-ART resistance testing over the no change, and the dolutegravir use even more so compared with the no change. If you want to see the within setting scenario differences between these, those are down the bottom. And there's comparing these means, there's almost no stochastic variation in these results, but there is a variability between setting scenarios. And then how, within the context of this model then, that models viral load and how that affects CD4 count and risk of death is informed by the latest CD4 count, the latest viral load, age, and whether people are on treatment, etc. And so how does that play out within our model in terms of the prediction of the death rate on average over the next 20 years according to those policies if they're kept up for the 20 years. And this is the death rate in people on ART. And again, the same format as before. If you look at the overall average, suggests that in particular the dolutegravir transition could be associated with something of the order of 1 per 100 person year reduction in death rate. And again, down the bottom you can see the within setting scenario variability. And you can see the 95% confidence intervals are very tight. So then what about costs? So I'm not going to give you all the costs here, but we made the, I think, somewhat now conservative assumption that dolutegravir is $44 a year, so slightly more expensive than efavirenz, when it seems like it may well turn out to be the case that it's the opposite. Tazanavir is assumed to be used in second line at $213, and the cost of a pre-ART resistance test is taken as $100. But if you look at the breakdown of costs according to those 3 policy options, you can see that you can probably just about see that the dolutegravir first line actually overall is leading to a lower cost. And that's driven by the orange band there, which is the cost of second line. So because of the reduced accumulation of drug resistance first line, it's predicted to be less use of more expensive second line regimens. So even though dolutegravir itself here is assumed to be slightly more expensive than efavirenz, overall it's predicted to lead to slightly lower costs, overall program costs. And now showing the sort of cost effectiveness plane, which on one plane shows on the x-axis the DAL is averted, so the health benefit, and on the y-axis the increment in annual cost. And so you can see, compared with the no change at the origin, that that both of the other policies are predicted to avert DALIs. The pre-ART resistance test leads to a slight increase in costs, but as pointed out before, the dolutegravir first line is predicted to lead to something of a reduction in costs. And so that would imply the policy option of choice amongst these three certainly would be the dolutegravir first line due to the lowest cost and the highest DALIs averted, within the context, at least for now, of the more than 10% of ART initiators having NNRTI resistance. But now moving to a situation where we're no longer assuming it's more than 10%. I'm now breaking down on the y-axis here, breaking down the 2017 level of pre-ART NNRTI resistance from 0 at the bottom up to the highest above 27.7% in some setting scenarios. And then on the x-axis shows the predicted reduction in death rate in people on ART if the transition is made to dolutegravir in ART initiators versus no change. And you can see that at the bottom there that even where there's no resistance, pre-ART resistance to efavirenz, there's predicted to be some benefit of dolutegravir, which basically follows from the trials which show benefits of dolutegravir even in the context of no baseline efavirenz resistance. So a few comments. I didn't have time to show you sensitivity analyses where we considered a kind of worst plausible case for dolutegravir, but it didn't change these results very much, but it was taking into account things like the risk of iris potentially raised with dolutegravir, taking into account that maybe the risk of resistance is slightly higher than we had assumed, and assuming some increased risk of virologic failure, perhaps due to the interaction with rifampicin, which might in reality not always be taken into account properly. Second point here is that high levels of pre-treatment drug resistance should prompt review of ART program quality in any case. To point out again that we focus on low-income settings in sub-Saharan Africa, and as Santiago explained, elsewhere generic dolutegravir is unavailable, and so this whole dynamic and this whole question doesn't need reworking for different settings. There's few data on safety so far in pregnancy of dolutegravir, although there's a few presentations here at the meeting, in particular one from Botswana, which don't seem to be throwing up alarming issues. The dolutegravir dose, as we know, may need to be increased with rifampicin, and that is the recommendation to double dose. The generic fixed dose combination with dolutegravir, I believe, is not available yet, but should be available within 2018, I understand. And just to point out that these results were is kind of an edited version of results that were presented to the WHO Guidelines Development Group on the public health response to pre-ART HIV drug resistance. And we're talking here generally about antimicrobial resistance. And just to point out that in principle, the approach used here, the cost effectiveness approach, the DALIs averted, and comparing different policies and the increment, that exact same approach ought to be able to be used to inform policy relating to antimicrobial resistance more generally, albeit that in those other areas, the policy options will often not include use of new effective drugs that we have, like dolutegravir. So the conclusions are that a future transition from efavirenz to dolutegravir is predicted to be effective and cost-effective in low-income settings in sub-Saharan Africa, actually regardless of the level of pre-ART and NNRTI resistance. Timing and exact nature of the transition, though, will depend on several factors. And again, these factors have really nicely been pointed out in the WHO document that was also released this week on transitioning, the issues with transitioning to new regimens. But certainly the level of pre-ART and NNRTI resistance is one of the factors to consider. And then just to point out that the consensus statement in the guidelines, if you haven't seen it, was that countries in which the prevalence of pre-ALT, HIV DR and NRTIs amongst people initiating first-line ART, regardless of previous ARV exposure, is more than 10%, should urgently consider an alternative first-line regimen that does not contain NRTIs, as defined in the 2016 WHO Consolidated ARV Guidelines. And in adults, that really means dolutegravir, I would say. I'd like to acknowledge all of the members of the working group who contributed to this, and some funding from WHO and the Bill and Melinda Gates Foundation. Thank you.
So hold on to your questions, because that's a lot of information about cost effectiveness, and how these models helped inform some of the guidance that, that we've made at WHO. And now we're going to move into the next part of the session, when we're going to start to talk about how what we've learned and what we're presenting in HIV drug resistance can be linked to the broader antimicrobial resistance agenda, as well as TB and malaria. So it's my pleasure to introduce Professor Dinan Pillay, who will be presenting on what is the connection between HIV DR and the broader AMR agenda. He is an academic clinical virologist and with a PhD in metabolic biochemistry and specializing in virology. I can skip all of that too. The fact that he's now in South Africa at the leading a Durban-based consortium of the KwaZulu-Natal Research Center and the TB KRIF, and also the Africa Health Research Institute, where it's become that now, with all of the work related to both TB, HIV, population studies. So he's in a very good position to give us some more information about how he may be expanding also to AMR.
Thank you, Megan. Thank you for the invitation to talk. So, yeah, I'm going to touch now on as you've heard, take this into the AMR discussion, and I'm not sure to what extent the audience is universally familiar with this, but antimicrobial resistance as a term is now really heightened in policymakers' minds, in governments' minds, and is taken to mean antibiotic drug resistance. But of course, all pathogens, whatever form or that they are, have the capacity to develop resistance to drugs that they're put under selective pressure from. And so I intend to just cover some of those issues and end up by arguing that there's far more similarities than differences between antiretroviral drug resistance for HIV and AMR in general. I am an HIV virologist, so I've shamelessly borrowed slides from colleagues on other areas, and just please bear with me in terms of my presenting those data. So you've seen an updated form of this this data set, and I wanted to go and just highlight on this older version the top left-hand figure, which is around the time of HIV drug resistance emergence in East Africa, but as a function of years since rollout rather than calendar year. The importance of this is it demonstrates that there is a temporal relationship between the starting to use a specific antiviral, in this case, and emergence of resistance. And we've got to consider that in the context of antimicrobial resistance in total. So please remember that. And of course, as you see data on transmission of resistance over calendar time, one's got to consider when in that particular country the rollout started. So in other words, what selective pressure was brought to bear on on that particular virus. But now AMR has become a key topic of policy. Both the WHO as well as governments around the world are developing policies and protocols to limit antimicrobial resistance. And this slide summarizes some of the salient characteristics here. As I mentioned, all pathogens will have the capacity to develop resistance against drugs against them. And these resistant strains survive and they can transmit. There are two broad mechanisms by which this happens. Within HIV, we're very familiar of the genomic resistance, the ability of the virus to generate variation in its genome, and therefore for there to be a selection of resistant mutants. And that is common for many pathogens. But of course, there's also the ability of plasmids to carry resistance between bacteria, and this is particularly relevant when we come to talk about, for antibiotic resistance, the One Health agenda, which is the interaction between animal health and human health with regard to antibiotic use and transmission of resistance. And these types of antimicrobial resistance cover bacteria, cover viruses I've talked about, cover parasites, and cover fungi. So this really is a major threat to human health in total, and I think it's very well worthy of the focus that there is now on this. So just to touch on some non-HIV pathogens, here's something that describes the global TB drug resistance epidemic. And this slide, if you look at the sort of more orange-colored parts of the map, it identifies those countries that have had access to bedaquiline as either on a named patient basis or expanded access for treatment of MDR and HIVDR TB. And one can see that this is really increasing around the world and is a crisis for TB management. And as I'll come to talk about, there really is a paucity of new compounds coming out to deal with this, this epidemic. Another pathogen, malaria, and you'll hear more about this, but this just demonstrates, again, resistance and patterns of resistance across the world of where malaria is in existence, both in terms of chloroquine resistance, sulfadoxin pyrimethamine resistance, and the artemisinins resistance here in blue. And note that this at the moment is in Southeast Asia, but there's all expectation that the artemisinin resistance will again spread to Africa. So this, just picking a few organisms, demonstrates really the global nature of and the ability of drug resistance to spread. So putting this into context, I wanted to show two slides from the O'Neill report, which was commissioned by the UK government in 2013 and reported in 2014, which looked at trying quantifying the global risk and determine the agenda, at least of the UK government, to deal with this. And this report estimated 700,000 deaths due to AMR now, with the risk of, by 2050, 10 million deaths per year due to AMR resistance, with, of course, the vast majority of those deaths being in low and middle income countries. So again, demonstrating that these infections know no borders, and therefore the adverse effects of antimicrobial resistance also know no borders, and it's a challenge for us all. So a little bit more detail now on a couple of those infections. This is a complicated slide, but demonstrates a lot of the characteristics of, in this case, TB and the evolution of XDR TB. If one looks on the very right-hand slide at the top corner, you can see Tugela Ferry. That is the area of KwaZulu-Natal where XDR TB was first identified. And there are genetic characteristics of the TB bacterium that will confer this XDR TB characteristic and phenotype. And what this slide shows, working back to the left-hand side, is after whole genome sequencing of a number of isolates of TB from this region of South Africa, this province of South Africa, how this extreme drug-resistant clone has emerged. And there are a number of lessons here which have parallels with HIV. So if one goes to the left hand, and this is a phylogenetic tree, so the tips of this phylogenetic tree represent the different bacteria that have been sequenced, TB strains that have been sequenced, and then applying statistical methodology and a molecular clock, it's possible to work back in time and estimate the origin of each of these resistant species. And what this demonstrates, and here is the color coding of the drugs available for TB, and also down here, the timing at which each of those drug classes were introduced in South Africa. And basically this demonstrates that going from single resistance to single drugs and then multiple drugs, there's been a continual evolution of each of these modes of resistance to end up at the top here with this cluster of bacteria that confer extreme drug resistance. And so there's a close relationship between the timing of introduction of drugs to the emergence of resistance, and then that resistance continues to aggregate new mutations associated with resistance. So immensely worrying and demonstrating that we're sitting on this around the world as we speak. With regard to malaria, this is the mutations associated with artemisinin resistance. Here, it's limited at the moment to the Thai-Cambodia, Thai-Myanmar borders, and here there are different sorts of mechanisms by which the malaria parasite generates resistance. But of course, this is a major problem given the limited number of therapeutic agents, and you'll hear a little bit more about that. So where does this lead us in terms of drug development and how we should be thinking and planning for future drug development? And again, I'm going to touch on different organisms and different types of organisms, starting here with antibacterial drugs. And this golden era of antibiotic drug development from the 30s into the 50s in particular, and then look at this complete void of discovery in the last 30 years. And again, this talks to the need to incentivize industry, incentivize biotech to continue to develop drugs, particularly for the gram-negative bacteria for which resistance is widespread. With regard to TB drugs, I showed you before the relatively small number of TB drugs available, and this gives the current state of play with regard to new TB drug pipeline, but there's very few drugs in that late stage of development that will be available now for the XDR TB bacteria circulating. And contrast that to HIV. Here is the already approved HIV medicines, and this is FDA-approved medicines, and you'll all be familiar with this story from Zidovudine in the 80s through to other nucleosides, non-nucleosides, protease inhibitors, and so forth, right till the current day where there's still new approvals, although I should say latterly this is focused more on new formulations rather than new drugs. But nevertheless, even if we look at 2017, there's a number of new forms of antiviral agents of different types that are being developed. So HIV already has a large amount of drug options, but of course, as we know, these are expensive and very few countries have access to all of these. So just what is the response? And I'll just show a couple of slides of the South African government response that is been based really on WHO guidance as well, is developing, in this case, a ministerial advisory committee on antimicrobial resistance, including a number of stakeholders in recognition of the importance. And the pillars of this AMR strategy are really around diagnostic stewardship, stewardship of use of antibiotics and to limit the emergence of resistance, surveillance, increased diagnostics, and also infection prevention. Because, of course, transmission of drug-resistant species of bacteria or viruses are a function of transmission, and therefore reducing transmission is always a key component of limiting the burden of antimicrobial resistance. So just coming to the end, what does this mean with regard to similarities and differences between HIV drug resistance and AMR? Well, I think the similarities really outweigh the differences, and we should start to be thinking of this all as one. So first of all, these are global problems. Secondly, surveillance is essential at a global level. Thirdly, as I've just mentioned, transmission of resistant species of organism are a function of that transmission. And if one reduces transmission, one reduces that risk to human health. And that's just the same for HIV as it is for TB or malaria. Stewardship is an important component. This term has come up with the antimicrobial, the AMR agenda, but again is very relevant to HIV. This is oversight, good training of healthcare staff, the availability of good monitoring processes and being able to act on that monitoring of infection and treatment in order to minimize resistance. And we all know how important that is with regard to HIV. Of course, the silence refers to that these infections are silent. They don't immediately tell you that this individual is infected with a drug-resistant strain of pathogen, and therefore we need to have our diagnostics optimized, and therefore requires vigilance. And of course, there are different drivers of resistance. These are different organisms that replicate in different ways, and we need to take that into account in terms of how monitoring happens. I've told you the R&D differences. HIV, there's been far more R&D in basic understanding of viruses, of HIV, which has therefore led to new development of antivirals. And of course, the One Health issue, which relates animal health to human health, which is clearly not relevant for HIV. And just finishing off about AMR stewardship, I would like us to coin this phrase for HIV. And basically, what this means is, as viral load monitoring is rolled out, it's essential we use that appropriately, because the more viral replication that goes on in the presence of drug, the more resistance that comes up. So really, stewardship of antivirals for HIV is an essential component if we're going to limit the ongoing spread of HIV drug resistance. So I'd just like to thank my colleagues who've shared slides with me and you for your attention. Thank you.
Okay, thank you very much. And now we'd like to pass along to our last speaker. And I know that this is the hour of the day where things become where everybody gets tired. So we hope you can stay to the very end. But we are very pleased to have Magda Robala Silva speak to us. She's the Communicable Disease Director from the Afro region of WHO. And she is in charge of looking over AMR as well as TB, malaria, HIV, hepatitis, and STIs, among many other things, for the African region. So, our pleasure.
Thanks, Meg. Good evening, everyone. I know it's not the best time to be speaking, but I want to assure you that even though it's the last presentation, I'm going to talk about where things actually do happen. How and what should the HIV program learn from TB and malaria drug resistance programs, because the malaria and the TB programs have been dealing with the drug resistance issue for a long time in large, massive populations, and we are going to see what can we learn from them. Most importantly, it is important to learn from them because we are dealing with the same countries, we are dealing with the same health systems, with the same communities, the same districts, and in many cases, the same people. We need to make sure that we optimize the systems that we are employing to deal with drug resistance. I have no conflict of interest to declare, and I will take you quickly through a couple of slides, starting with the problem of drug resistance. It has just been defined by the speaker who was here just before me to say is a natural mutation, is a natural event, it's inevitable. But a lot of it also happens because of the things that we do or the things that we fail to do. Inadequate treatment regimens, stockouts of medicines, the quality of care, interrupted regimens, poor adherence of individuals to the regimens that they are prescribed to treat the diseases, poor treatment monitoring, but also because the prescribers sometimes fail to take care or pay attention to social or cultural factors that lead to poor compliance and poor adherence of individuals to the treatment regimens that they have been prescribed. If I look at HIV drug resistance, you have seen this slide before. This is a worldwide map that shows where the HIV programs have been doing surveys, national representative surveys, to try and map the prevalence of HIV drug resistance. Yesterday, you also, those of you who attended the launch of the Global Action Plan and have seen the HIV drug resistance report, you have seen that out of 11 countries that have nationally representative data, 6 of them have reached and have gone above the threshold of 10% of individuals on, as ART initiators who presented with drug resistance. Out of those 11 countries, we have 2, Nicaragua and Uganda, which have actually above 15% resistance among people who are ART initiators. This is a situation that will continue to evolve as the HIV program continues to document the situation of HIV drug resistance across the globe. If we look at TB, the MDR TB surveillance is a quite well-established system. They have a global coverage of surveillance data on drug resistance that spans for over 20 years. And you can see, for example, the areas in orange, that's where there are ongoing surveys from last year, 2016, and this year we'll be able to have data from those countries. 155 countries in the world are have surveillance systems for drug resistance tuberculosis, and 83 of them have continuous surveillance systems. But if you look, for example, the white areas, mainly in the African continent, these are places for which we have no data whatsoever. So there is still a lot of work to be done in terms of mapping the situation of MDR TB. There is a special report that has been published by the New England Journal of Medicine, which, if you have not seen, I would like to encourage you to go and read, because it gives a very good analysis of the work that has been done by the TB program in terms of documenting the drug resistance TB across the past 20 years. This slide is intended to show you the MDR TB cases among newly diagnosed TB cases. And you can see this area, which is dark brown, where you have over 18% of patients which are newly diagnosed with tuberculosis but who have multi-drug resistance tuberculosis. Again, if you look at the map of Africa, you still have many areas with no data on MDR TB. This red map is to show the situation on XDR TB. 118 cases as of October last year have reported at least one case of XDR TB, and 9.5% of those with MDR TB cases have extensive drug-resistant TB. So the situation is not as good as you certainly know. Moving to malaria, you have seen this slide as well. You all remember late 2000, the entire world had to shift to artemisinin-based combination therapies. We thought we had found the solution to the failing chloroquine and SP that has been haunting malaria endemic countries. We thought that artemisinin-based combination therapies will be with us for much longer time than it turned out to be. The Southeast Asia Mekong area in 2008, starting in Cambodia, told us that unfortunately artemisinin derivatives were not going to be with us for too long for treating malaria. It is a matter of time before in the African continent we start seeing malaria-resistant cases to ACTs. This slide is to show what the situation currently is in terms of ACT resistance in the Mekong sub-region. The red on your left-hand side, the red dot is Cambodia, which has resistance to 4 artemisinin-based combination therapies. The blue countries are resistant to 1, and the pink countries have resistance to at least 2 ACTs. On your right-hand side, the green map is depicting the same situation, but the darker the green, the worse the situation in terms of resistance to a number of artemisinin combination therapies for treating malaria. So against this scenario, what have been the responses of the different programs, the malaria program, the TB program, and also the HIV program, and what are the lessons, what are the common lessons that we can learn, what are the challenges that we still have and that we need to address if we are going to protect people and protect some of the medicines that we are still have and that can contribute to reducing mortality and helping us achieve the 2030 SDG goals. For HIV, you have been in this conference since yesterday. The program has launched the Global Action Plan on HIV drug resistance. There are 5 major points for dealing with that. I will not dwell much because my predecessor has explained in detail what the prevention and response, monitoring and surveillance, research, innovation, lab capacity, and governance elements are meant to represent to the countries that are grappling with the problem of HIV drug resistance. I just want to mention that this Global Action Plan is in line with the Global Action Plan for Antimicrobial resistance. You have seen this slide. I will just continue. I wanted to let you know that some countries, many countries have started in detail looking into surveillance elements to be able to address the issue of drug resistance. The previous slide of the 11 countries with national representative data, you have seen Namibia is one of them. They have 249 ART sites where they do monitor the quality of care. They have early warning indicators that are used to take action as soon as necessary in order to address the issue and prevent HIV drug resistance. Some of the indicators are related to whether or not patients come on time to pick up the medicines, how long are they retained in care for at least 12 months, whether pharmacies have stockouts of medicines, what are the dispensing practices, viral load suppression, etc. If we look at what the TB program has been doing in terms of addressing, preventing drug resistance, the TB program has been collecting data, like I said, over the past 20 years, and they have a system which is quite well established, and they collect annual data around March and May from close to 200 countries, and they have quite a robust system to monitor TB drug resistance. These different documents are the global TB reports that have been produced, and in detail they give you the situation for countries for which data is available. They have been producing guidelines in order to help countries monitor and handle their TB drug resistance challenges. 5 priority actions promoted by the TB program in order to deal with the programmatic challenges of drug resistance TB, infection control, high quality treatment, expanding testing and detection of drug-resistant cases, and political commitment and financing for the programs. Moving to malaria, there is also a well-established algorithm for making a decision on whether or not it's time to change the first-line antimalarial medicine. The threshold, again, similar to the HIV, is 10% for failure of the first-line anti-malarial medicine. The Mekong, Greater Mekong sub-region is a particular case of ACT resistance. There has been an emergency plan for containing the ACT resistance, and countries in the Greater Mekong sub-region are now moving into malaria elimination as a way of addressing the source of the problem. WHO has been working with them in collaboration with the partners. The emergency plan was meant to increase coordination, the quality of interventions, and contain the spread of resistance to ACTs. So what, from these 3 programs and the experiences that we have seen over the past few years, what are the missing pieces that we think the different programs need to address? We think the issue of routine surveillance continue to be a missing piece that needs to be addressed. We need to have a better understanding of what the prevalence of HIV drug resistance is, but the same applies to TB and malaria. The awareness on the threat that is posed by HIV drug resistance still needs to be well and better understood by the countries. We still have gaps in knowledge of HIV drug resistance in groups of populations. We need to take action to address quality of care, quality of treatment, quality of services, reduce loss to follow-up, improve adherence and retention, limit or eliminate drug stockouts. Capacity of laboratory diagnosis in our countries is a major problem, more in some than others, but definitely an issue that needs to be addressed. And most importantly, any data that is available needs to be used for action. TB, HIV, and malaria have established thresholds for which, when it has reached a shift to a second-line medicine, needs to promptly be done. In summary, what did HIV programs learn from the TB and malaria programs? That there is a need for a clear strategy and plan of action. I think with the launch that we have seen in this conference, definitely the HIV has applied one of these lessons. Country ownership is important. No matter how well the strategies and the plans are defined, if they are not owned, adopted, adapted, and utilized by the countries, we will not make progress. We need strong surveillance systems and, most importantly, use the data from the surveillance systems to take action to trigger the response based on the threshold that has been defined. The actions need to focus on the people, on the clients, on the patients. We need to make sure that the barriers to adherence and the concerns that the patients have are addressed if they are going to collaborate with us, because we really need the patients to adhere to treatment. We need robust laboratory networks and, of course, partnership and funding. I will end with this slide, which is from the TB program. I thought it is a very good summary of some of the lessons that we all need to be applying. Partnership is key, and partnerships is with patients, with communities, the ministries of health, partners, the donors, etc. But also partnerships will help us with capacity building, advocacy, guidance, and we need to share what we know, what we have learned, so that we can be better and stronger together. Thank you for your attention, and I want to thank the entire WHO team who has provided information for this presentation, the Ministry of Health of Namibia and the University of Namibia. Thank you.